Hello, and welcome to this Nature.com custom webcast titled Defining Physiological Normoxia in Cultured Cells for Translation to Animal Models and Man. Today's webcast is sponsored by BMG LABTECH, who are a leading global developer and manufacturer of innovative, high-quality and reliable microplate reader instrumentation. We'll begin today's webcast with presentations from Catherine Walk, who is an application specialist at BMG LABTECH, Dr. James Hines, who is the Head of Research and Development at Luxil Biosciences, and Professor Giovanni Mann, who is a Professor of Vascular Physiology in the Faculty of Life Sciences and Medicine, King's College London. We will end with a Q&A session where we will address the questions that have been submitted by you. To ask them a question, just type your question in the Ask a Question box and press Submit at any point during the webcast, and we will answer them today. And now over to Catherine.
Thanks, Jason. Okay, everybody, thanks for joining us. My name is Catherine Walk, and I'm an application scientist for BMG LABTECH. This group webinar was planned to present a universal problem for cell-based biology research, that of what is normoxia and the correct conditions at which to test cells, and to try and present some potential hardware and reagent solutions to aid in resolving this issue for microplate reader assays. So for my part, I'm going to be presenting the CLARIOstar microplate reader from BMG. This will be used for all the research that both James and Giovanni will present in their following presentations. The CLARIOstar is a multifunctional microplate reader which uses cutting-edge technology such as our patented LVF monochromator, and also allows users to create bespoke physiological conditions for their assays. So to talk about the CLARIOstar as a microplate reader, it allows you to detect all non-isotopic read technologies. We use different optical systems to provide the best performance in all assay technologies, with a linear variable filter monochromator for scanning and bespoke assay design for fluorescence and luminescence. We offer filters for the most sensitive assays, such as time-resolved fluorescence and fluorescence polarization, with the ability to mix and match with the monochromator. And we have an ultra-fast spectrometer for absorbance and even a laser for alpha screen technologies. So to go further into the features of the CLARIOstar before we start to talk about atmospheric control, and pay particular highlight to the linear variable filter monochromator, I'd like to discuss that as it offers near filter-like sensitivity in a flexible wavelength scanning device. Users can both define the wavelength of interest, but can also vary the bandwidth of the monochromator from a range of 8 to 100 nanometers. This range and feature is unsurpassed by any other system and gives users really the freedom to develop assays and to reproduce published filter settings as required. The ultrafast spectrometer allows users to measure multiple wavelengths or a full spectra, for example, for nucleic acid detection, and offers spectral outputs in under 1 second per well, making it incredibly fast for users. The system offers top and bottom reading as standard, and the air transport optics are particularly important for cell-based assays from the bottom read. We achieve fantastic performance here, and as you can see from the representations at the bottom of the page, we offer different read options for the most accurate and reproducible detection of non-homogeneous cell layers. So, with respect to injection, or if you like, stimulating cells, for example, for calcium or enzyme-related assays, the system can be fitted with 2 injection systems onto the reader. This reagent-saving system allows users to control volumes added to each well and also the speed of addition, which can be important when injecting onto a monolayer of cells, whether using the atmospheric conditions or not. So speaking of atmospheric conditions, now we go to talk about the use of our atmospheric gas controller. BMG have a long history in this area. Customers have always pushed us to produce more in the way of atmospheric control other than temperature control. And it's really driven by many users now using specialized incubators or hypoxia chambers, for example, to try and induce phenotypes in their cells. Previously, users would have needed to put their microplate reader inside such a chamber, and having the ability to control the gas in the instrument means you save space in your workstations, and also you can continue this idea of working under physiological conditions throughout all of your work. From the year 2000, we've worked with mixed gases, and it's really gone through CO2 to independent control of oxygen and CO2 to the CLARIOstar we have today, which offers a computer-controlled system. So the system we have on the CLARIOstar is a standalone module that sits on top of the CLARIOstar. It can be controlled through either a touchscreen or through the CLARIOstar software itself. And the gas levels can either be displayed on the unit, but they're also shown within the software and also recorded with the data analysis software as well. Gas levels can be regulated by the user from 0.1% to 20% for both oxygen and CO2. 100% nitrogen is used to purge out and control oxygen, and 100% CO2 is used to regulate the CO2 levels in the instrument. Users can program up to 10 presets, and the system is completely calibrated, and gas usage and real-time monitoring can also be viewed through the display panel. There is even a correction for altitude, so as this can also impact on gas control depending on where your lab may be based. So this device enables a broad range of assays to be performed, including metabolism, as we'll hear from James at Luxel, Ross, as we'll hear more from Giovanni's research, and many other cellular markers that can be detected in long-term assays, as these assays are increasing a lot now more over time. So this actually goes on to show, I go on to show here an example, if you like, a proof of principle of the gas control in general. This is also a progression of how microplate cell-based assays have evolved. So this assay format from Promega offers a non-destructive assay measuring both proliferation and toxicity. It can be monitored kinetically in cells for up to even 4 days in culture. So CellTox green is a fluorescence readout for toxicity. The dye is quenched when it's outside of a healthy cell, and only fluoresces when it gains access to a dying cell. The real-time glow is a luminescence output of proliferation. This measure is based on the metabolism of an empty substrate by the cell, and the luminescence will increase as cell numbers increase. A non-viable cell would not metabolize the substrate and therefore wouldn't give any light. Both approaches, as they're using different read modes, can be multiplexed by the CLARIOstar and detected in real time for both assays. So to show some general assay here, again, proof of concept of the gas controller. Most people historically would have used 5% CO2 but atmospheric oxygen. So in basic terms, proof of principle of how the gas controller works. We can see from the red line that proliferation increases over the 72-hour period that was used for testing, and the blue line in the top graph represents the toxicity. So we can see that the cells remain healthy when growing at CO2 in atmospheric oxygen for the 72-hour period. And the graph below would show the use of a compound treatment where growth is inhibited at higher concentration levels and toxicity is seen. So the top part of this lower graph would be toxicity and fluorescence, and the bottom part would be luminescence, showing the proliferation results. So taking this further, we've talked about, and we're going to go on to talk about more about oxygen control, and really driven by the interest of users measuring HIF or cancer, ischemia, reperfusion, we have developed our ACU further this year to also incorporate the reintroduction of the atmosphere to previously oxygen-deprived cells to assist in modeling different disease states. Now, what you see on the screen is quite extreme, from 18% to 1%, but just to show really the possibilities that are there with a system like this. So in very rapid time, we can both drop the level of oxygen within the cell. Within the well, sorry. It's even possible to regulate the CO2 during this period. And within very short periods of time, even less than 25 minutes, we can take a well from being at 1% oxygen back to 18% oxygen. Now, moving further with this data, and introduces in some ways how we've been able to work with LUXCell. BMG LABTECH are partnered in a grant, Horizon 2020 grant called MetaCell, with both LUXCell and Cardia and some strategic academic groups in Oxford and Imperial. This MetaCell grant looks at a gold standard metabolomic solution. This data here just shows the concept, takes it to the next stage, shows that the O2 probes from LUXCell can validate the oxygen levels that are induced by our instrument in terms of the purple line and the grey line tracking each other, as we see on the screen. There are, of course, differences between the level of applied oxygen and that of the respiring cells, which is more a question for James and Giovanni to answer today. But from my side and the CLARIOstar side, I just wish to show how it's possible for users to create multiple readouts in real time from the same wells again at different oxygen tensions, as shown by the data to the right side, which the MMP, mitochondrial membrane potential, and ROS data that we can see in the graph to the right side. So in summary on the system, I can really surmise with the CLARIOstar, it's a versatile instrument, highly sensitive, with this gas ramping capability to maintain for prolonged periods, exacting gas tensions, and even perhaps offering the possibility to study things like ischemia reperfusion, moving from very low oxygen levels to more of a normoxia type environment. And lastly then I'd like to thank the people that have been responsible in producing the data that I've shown you today. And at this stage, I will pass you over to James to talk more about the oxygen sensing technology that I showed in my presentation. Thank you, James.
Thanks very much, Catherine. That's really interesting, and it's really kind of powerful technology that we're also using to try and help researchers to, as we've said at the start, try and narrow the gap between the in vitro model and the in vivo situation. So, as mentioned at the start, my name is James Hines. I am from LUXCell Biosciences. I'm head of research and development here, and we are a company focused on the development of advanced fluorescence-based technologies with a particular focus on oxygen consumption and, well, oxygen sensing and pH sensing. And for a long time, we've had a history of developing metabolism assays with an initial focus on mitochondrial function and moving from there to cell metabolism. And we're now developing and providing solutions to measure cellular oxygenation for application in these types of areas around hypoxia and normoxia because of the importance of that parameter and how we feel it will improve the physiological relevance of the in vitro models currently being developed. So if we take a look in the first instance at, I suppose, the people that we're working with to develop those types of solutions, as Catherine mentioned, we're part of a Horizon 2020 project with some key collaborators, both cell model providers within Cardia and advanced instrumentation providers with BMG, and some key, as Catherine mentioned again, some key academic collaborators in King's College London, or in Imperial College London and in Oxford. But also to mention the long-term collaboration we have with Professor Giovanni Mann on the cutting-edge work that he's doing in demonstrating the key importance of both preconditioning cells at physiologically relevant oxygen concentrations and the biological impact that can have on the experimental outcomes. And we'll hear much more on that later. Now, just to maybe take a little step back and take a look at metabolism in general, some listeners may be more familiar with this than others. So to recap a little bit, if we take a really simplistic analysis of cell metabolism and we take ATP being produced either via glycolysis or oxidative metabolism, glycolysis can be measured by assessing the rate at which the cell is acidifying the media that it's in, whereas aerobic ATP generation can be interrogated by assessing the rate at which the cell is consuming oxygen. And we have developed a suite of assays to probe these aspects of metabolism. We have a set of assays to measure extracellular acidification or glycolytic flux, and then stress tests to assess glycolytic capacity, and also assays to measure oxygen consumption, mitochondrial capacity, and other key features or parameters of metabolism, including fatty acid oxidation. Now, these are all plate reader assays, and as Catherine mentioned, we've done a lot of work on the CLARIOstar to make them as convenient as possible. One of the key parameters and the core parameter for today's discussion is around oxygen concentration and the significant impact oxygen concentration can have on these metabolic pathways and on other key signaling pathways. And the solution we've developed for monitoring intracellular oxygen or cellular oxygenation is the MitoXpress-Intra oxygen or intracellular oxygen assay. And that's the main technology that I'm going to describe today, and maybe describe the area around which it's applied. So if we take a look at the objective of much of this research, it's to improve the physiological relevance of the in vitro models that are typically deployed in biology to give us a better picture of what's happening in vivo. And there are a lot of very, very innovative companies developing 3D models and cell models to try and advance this whole field, whether it be through human iPS-derived cell models or spheroids or particular other 3D approaches to this. And significant strides have been made in that space, and they're growing in sort of application across biology. One of the key aspects of this analysis, however, is oxygen concentration, because of the difference between oxygen concentration applied and oxygen concentration that the cells will typically see. So if we take a look in the first instance and assess the oxygen concentration that cells are typically cultured at against the oxygen concentration that tissues typically see. So oxygen concentration in tissue culture is typically at about 18.6%. That's because of percentage CO2, etc. Now, if you compare that to the oxygen concentrations that the tissues typically experience, it's significantly higher. So tissues typically see oxygen concentrations of between 10% and down. So, microelectrode measurements of kidney can be at 10% or below, liver can be at about 5%, and muscle can be far lower. And again, Giovanni will go into far more detail in this area. But what that points out is that this idea that hypoxia is simply a measurement at 5% oxygen is not really relevant to the in vivo situation. And it points to an area of normoxia or physoxia around 5%, again, tissue dependent. But more importantly, it highlights that the oxygen concentration that much of these in vitro models are being applied in. One of the key areas in all of this is trying to narrow the gap between the in vivo situation and work that's happening on the bench in vitro. And a lot of innovative companies have developed new approaches to this, developing new technologies, new cell models, whether it be human iPS-derived cell models, spheroids, or other 3D structures. However, one of the most important aspects of ensuring that your cell model is close to the in vivo situation is to be very cognizant of the oxygen concentration that these analyses are being performed at. And if we take a view that cellular culture is typically performed at 18.6% oxygen, and we compare that to the oxygen concentrations that tissues typically experience, it is significantly higher. So the kidney experiences an oxygen concentration of about 10%, obviously varying depending on where that's measured. These would be from sort of microelectrode-based measurements. Liver would be lower, muscle would be significantly lower again, and Giovanni can speak to this in significantly more detail. But what this highlights in general terms is that the conditions under which tissue is typically cultured and measured is hyperoxic for most of these tissues. So if we bear that in mind, then it poses a particular question. What impact does that hyperoxic effect have on metabolism, have on cell signaling, and on the physiological relevance of the testing that's performed? And how can we try and improve that such that we can control and monitor the oxygen concentration, and we can probe the relationship between oxygen and metabolism and signaling? So we've already seen from Catherine the powerful technology that is equipped within the BMG CLARIOstar, where you can modulate the oxygen concentration in the measurement chamber. And Giovanni will explain how this can be incorporated with hypoxia workstations to give you an oxygen-controlled workflow. However, an important aspect of all of this is making sure that you're also monitoring the oxygen concentration, because it's important to understand what the cells are actually experiencing. And as I mentioned earlier in the presentation, our solution to that particular problem is the development of MitoXpress-Intra, which facilitates the measurement of intracellular oxygen concentration or cellular oxygenation. And we've worked with BMG LABTECH very, very closely to advance this particular technology to make it convenient for people to conduct these measurements. So you simply load the cells before measurement. That would typically be an overnight loading period. Wash the cells prior to measurement, and then you can conduct your analysis either in the presence of particular activators or compounds. Now, the compound or the probe itself spectrally is very, very powerful as well, in the sense that there's a big gap between where we excite the probe and where we measure emission. And that allows us to multiplex with other reagents, informing on other key aspects of metabolism. So in essence, what we have is a stable nanoparticulate oxygen-sensitive probe, which provides a simple loading procedure, is multiplexable with other key readouts, and importantly is plate reader compatible, so that we can do these analyses across 96 wells. And also importantly, from a biological perspective, is that we can output data in real time in oxygen scale. And in essence, what we're doing is we're monitoring the transition from a low level of signal to then, as we drop oxygen concentration, moving to a high level of signal. What that facilitates then is essentially a calibration function, which is already built into the CLARIOstar software, which allows us, as I said, to output in oxygen scale. And that is quite a powerful approach, because what I'm looking at here is a screen grab from the MARS software, the BMG data analysis software, and in the same well, at the same time, we're monitoring the environmental oxygen concentration in green, and we're monitoring the cellular oxygenation levels in red. And as you'll see on the left in column 2, the cell oxygenation level is mapping almost exactly the environmental oxygen concentration level, with a bit of a gap for diffusion, because these cells are not respiring. However, as we move across the plate and the level of respiration increases significantly, you can see the dramatic difference between the environmental condition and the intracellular level. It's this type of analysis that we are now able to provide. Now, if we look at that in greater detail, you can see that again with the atmospheric control unit, just to demonstrate the extreme levels that can be achieved, again, as Catherine said, going from high oxygen to low oxygen, you can see how those differences are quite profound. So what that does is, this ability allows us to question some key assumptions that are often applied in this area. And the first assumption is that all you need to do to achieve a particular oxygen concentration that you're interested in studying is to set your workstation or your instrument to that concentration. And these data demonstrate that that is not the case. So on the top line in purple, you're looking at the oxygen concentration in the measurement chamber as we step down that oxygen concentration. The bottom line is the oxygen concentration that the cells are actually experiencing. And here we're demonstrating a 6% difference in oxygen concentration just in these conditions alone. So again, demonstrating that it's important to monitor this particular parameter. Now, what that also allows us to do is to question the assumption that simply applying an offset allows you detailed control of oxygenation. So in this instance, can we just say that we set an oxygen concentration of 6% off what we're looking for, and there magically our cells will be at the right oxygen concentration? And the caveat that we would like to highlight to that is that metabolism is an extremely dynamic process. And in this example, we're looking at cells that are being analyzed at 8% oxygen. So the chamber, as you'll see in black, is at 8% oxygen. But by very, very slightly changing the substrates that these cells are metabolizing, the same cells cultured in the same way, one is on high glucose at 4% oxygen, one is on slightly lower glucose, and they're almost driving themselves to 1%. And that can be quite a significant difference biologically. And these are data generated by our collaborators in Oxford University, Colin Morton and Michelle Potter. Now, if we take a look at a slightly more extreme example, but also quite relevant biologically, where in this instance we're looking at beating cardiomyocytes. And if these beating cardiomyocytes are sitting resting at 14% oxygen, despite the fact that the chamber itself is at atmospheric oxygen. And if we block respiration, we can return that to atmospheric. However, if we increase the beat rate of these cells, we can increase their oxygen demand, and they then drive themselves down to far lower oxygen concentrations because metabolism has speeded up, oxygen consumption has speeded up, and back diffusion just can't feed fast enough. So these cells are sitting at 6% oxygen despite the fact that they're being measured at 21. So that's just highlighting the differences that can exist in this area. So it's not a valid assumption in all instances to assume that simply setting an offset achieves you the oxygen concentration that you were looking for. Now, to very, very briefly take a look at where this could be important, it's got a really important attribute in allowing you to delineate the relationship between oxygen concentration and other aspects. And taking a simple example, taking the example of tumor metabolism, if we take a look at cells that are being measured at atmospheric oxygen, these cells in particular, when you measure intracellular, are at 17%. We can monitor extracellular acidification in the same well at the same time as oxygen concentration. So at 17%, we have a particular level of glycolytic flux or extracellular acidification. But as you drop oxygen concentration from 17% to 5%, that increases dramatically as the cell becomes more dependent on glycolysis for ATP generation. And in areas such as assessing the therapeutic efficacy of, for instance, radiosensitivity, these types of analyses and measuring at the correct oxygen concentration can be critically important. This is a figure taken from a recent paper which is demonstrating the impact of oxygen concentration on this efficiency. So if you were testing for efficiency at high oxygen concentrations, you may be missing the fact that these particular compounds are not that very effective, or these particular treatments are not that very effective at lower O2. So it's important to, on the first instance, know what oxygen concentration you should be measuring at, and secondly, to ensure that you're actually measuring at those concentrations. And Giovanni will speak in much more detail to this later. And also, what is also critically important in lots of areas is to make sure that where it's required, that your cells are preconditioned to the oxygen concentration that you wish to be performing your analysis at. And that is a core feature of the research of Professor Mann's group. And again, we'll hear much more about that later. So to summarize, cells in vitro are typically cultured under hyperglycemic conditions, often because of the high glucose concentration, and hyperoxic conditions. This can have significant biological consequences, and technology now exists both to monitor and modulate the oxygen concentration. But that monitoring is really, really important, and that's a reason for us for developing MitoXpress-Intra. And what that means then is to be aware of the fact that it is not correct to just simply assume that by setting an oxygen concentration on an instrument, that is what your cells experience, and again, monitoring being critical, and also important to be cognizant of the contribution of varying metabolism to that oxygen concentration. And we feel that this can have a profound impact in improving the physiological relevance of in vitro models as they are developed, in combination with ensuring that the correct metabolic pathways are being recruited, and that the correct preconditioning has been applied, and that by multi-parametric analysis of oxygenation with other parameters, such as mitochondrial activity, such as glycolytic flux, on instruments that have the capacity to monitor these, such as the CLARIOstar, this could be quite a powerful tool for advancing this whole area. So with that, I'd like to thank Catherine for the initial discussion, or the initial introduction, and I'd hand over to Giovanni for the exciting aspect of the talk.
Thank you, James, very much. It falls upon me, one, to thank both BMG LABTECH and Luxel Biosciences, who have been critical in underpinning our R&D research in vascular biology here at King's. My role will really be to provide you with some data from our ongoing research, published and some unpublished, that defining the term normoxia, or physiological normoxia, in cell culture is absolutely critical for translation to interpreting studies in animal models, as well as clinical translation to man. So if I take you, please, to the first slide, we've had some of this information, and you'll see on the ordinate that the PO2 in kilopascals is shown, also in millimeters of mercury, but I'd like to equate kilopascals to percent oxygen. It's almost the same. And you'll see that at the top, upper left-hand part of this graph, left-hand part of the slide, we breathe in about 21% air, oxygen, and as we move down the alveolus, go to the small arteries, arterioles, precapillaries, eventually ending up in the capillary or microvascular bed where nutrients and waste products are exchanged, we're really sitting at an oxygen tension or level of somewhere between 3 and 7%. And in some cases within the cytosol, it can fall below 2%. On the right-hand side of this slide in color, I show this same concept again, where you have air at 21% kilopascals or 21% oxygen, moving to the lungs at 20, coming to the alveoli, noting immediately that we're sitting at 14% oxygen. And as we hit arterial blood going to organs in the body, it can vary between 5 and 13, and as low as 0.5 to 1.3% oxygen in mitochondria, depending on the mitochondrial state. I refer readers to an excellent review written by a close colleague, Professor Jeremy Ward, in BBA, published in 2008. My own work focuses on looking at human tissue, fetal tissue from umbilical veins or umbilical cords, and shown here in this graph, you will see in white, the y-axis is again kilopascals or percent oxygen from 0 to 5, and the right-hand side is oxygen in millimeters of mercury. If I lead you to the vein, which is the white column, you'll see that this bar gives you a mean value from many, many studies that we've researched in the literature of around 3.7% within the umbilical vein in vivo. In the artery is a bit lower, but for today's talk, that's not what I want to focus on. I want you to recall the 3.7% oxygen in the umbilical vein in vivo. To look at this at a cellular level, we isolate endothelial cells from this vein and we culture them in microtiter plates. And to recapitulate the in vivo physiological oxygen concentration, we have set up an R&D collaboration with Baker Ruskin. I'm illustrating this in this box that you can see at the top labeled Ruskin. It's called a cytive. This is a single cytive in which the interior environment is fully oxygen controlled, CO2 controlled, and humidified. Our cells enter through the little side port on the right-hand side of that box and are maintained in this box for periods of up to multiple passages in one case, or a minimum adaptation of 5 days. I've opened up the box on the upper right-hand side where you can see some cells actually being cultured. You can see a small white piece of instrument with a top that's a fluorescent microscope from Metaluma, very small. You see pipettes. So all the handling, treatment, trypsinization, feeding, experiments, and final obtaining of lysate is done within a defined oxygen environment. Once our cells are adapted within this environment, if we want to use the look at mitosocs to look at mitochondrial respiration or MitoXpress-Intra, as I'll go into to show you measurements of oxygen in living cells, we would simply transfer one of our 96-well plates into the BMG CLARIOstar oxygen-regulated unit, the plate reader. We have already established in our published work that this very rapid transfer of pre-adapted cells into the same oxygen level that they see in the cytive to the plate reader doesn't cause any reoxygenation problems. So really, it is the interplay between Baker Ruskin, Luxcel, and BMG LABTECH that enabled us for the first time to obtain real-time measurements of oxygen within cells. And in my next slide, what I'm showing you here is these are real-time measurements of medium and intracellular oxygen levels in fetal endothelial cells. These are fetal cells because the umbilical cord is fetal in origin. And if you look on the inset in panel A, you'll see oxygen content in percent from 0 to 25. Cells are cultured in room air, or effectively, as James already explained, it is approximately 18.6%. You'll see the open squares, rather small on this slide, denote the medium, and the black circles or squares below the cytosol in these living cells. And you'll note immediately that although the incubator is gas at 21% oxygen, we're achieving a medium level of around 18.6 or 18.9, and the intracellular concentration is lower. As we flip or change or lower the oxygen concentration within the BMG or CLARIOstar plate reader, you can see this very nice rapid decline, after about 2 hours achieving somewhere in the region at a cellular level about 3 or 4%. If, however, we look on the lower left-hand side of this slide, you'll see a simulated blood vessel. This is supposed to represent in vivo the umbilical vein, and there, with blood flow, we're achieving an oxygen concentration or content of around 3.7%, and the unstirred layer at the blood endothelial interface is minimal, so that the endothelial cells are actually, from publications in the literature, giving us a reading of about 3.7% oxygen. On the right-hand side of this slide, you'll see that if cells are cultured in air, that the intracellular oxygen content from the MitoXpress-Intra probe approximates around 18%. Now, you might assume that simply taking 3.7% and equilibrating the Baker-Ruskin citive at that level would give you the desired intracellular oxygen concentration. To recapitulate the in vivo level, the answer is, I'm trying to move my slide forward, excuse me. It is not. You'll see the blue data there, the blue bar, shows that if you have the chamber set at 3.7%, your cells are actually hypoxic. In other words, the intracellular oxygen concentration is below that dotted line, which is marking in vivo levels. Fortuitously, when we actually put our cells in a monolayer, that's the middle panel that you see there, you'll see that in the gas phase, setting at 3.7, actually you have a gradient of oxygen diffusion. So these cells are effectively seeing partial hypoxia. If, fortuitously, we put the cells at 5% oxygen, we ended up obtaining the exact level of oxygen that has been reported in vivo. So we're studying our cells under what we call physiological normoxia. Referees and reviewers have often asked us, as shown in this slide, this is our publication in the FASEB journal, it's online now, and they said, what is happening to HIF-1 alpha, the stabilization of the hypoxia-inducible factor HIF-1 alpha, well researched by Peter Ratcliffe and colleagues in Oxford. And you'll see in the upper immunoblot there in panel A, the oxygen percent from 1 to 18 on the right-hand side, and we're looking at HIF-1 alpha protein expression relative to the reference protein beta-actin. And you'll note that at 18, 10, and 5% chamber oxygen levels, we see no induction of HIF-1 alpha or stabilization, whereas it does come in at around 3 or 1%. And in the little graph below, where I'd like to draw your attention, the curve on the little graph below has oxygen tension on the x-axis and HIF-1 alpha protein levels relative to beta-actin on the y-axis. You'll see that if we just look at the black curve, that is the ambient oxygen in the Ruskin chamber. If we correct that for intracellular oxygen levels, we shift this to the left, immediately noting that the stabilization of HIF-1 alpha is actually only occurring at intracellular oxygen levels well below 4%. I'd like to skip what's on the right-hand slide for time constraints. The next thing that we can do with our endothelial cells is ask the question, if cells are adapted to 18% oxygen or 5% oxygen for 5 days, what is the protein content? Do they adhere the same way? Do they proliferate the same way? And these are umbilical vein endothelial cells cultured in medium 199 in our routine culture assays, and we immediately observed that their rate of proliferation was markedly lower in cells pre-adapted with a new phenotype at 5% oxygen. So we're affecting the phenotype of the cells. A reviewer immediately asks, are you not damaging proliferation of cells? No, we are not, and I'll give you further information on that shortly. In this slide, rather busy, please look at the immunoblots at the top. Here what we're looking at is taking umbilical vein lysates and immunoblotting them for caveolin 1, the first one, PP2A, that's a phosphatase 2A in middle panel, and calmodulin in the right-hand panel. These are the three key proteins that are upregulated in cells adapted for at least 5 to 6 days to 5% oxygen, achieving the intracellular oxygen level measured in the umbilical vein in vivo. I'd now like to turn to the use of the CLARIOstar and fluorescent probes such as FURA2. On the panel A, please stay with me with the umbilical vein endothelial cells in the 96-well plate. they have been loaded with Fer2AM as an indicator of intracellular calcium. What you will see is 3 oxygen concentrations, 18% black, 5% sort of darker gray, and 1% oxygen, very light gray. The bar at the top is histamine. The cells are challenged with 10 micromolar or 100 micromolar histamine. You'll see immediate store release of calcium and then a plateau influx. As we lower the oxygen in the CLARIOstar, having pre-adapted these cells for 5 days in the site of workstation, you will note a lower peak and also a lower plateau. The question, I don't have time to go into it, but it's in the papers if you look below, was, is this due to an alteration of influx of calcium following a vasoactive stimulus, or is it due to calcium reuptake or efflux across the plasma membrane? The right-hand side shows you an experiment where we do a calcium add-back. For those that know about calcium, this will be easy. If we stimulate cells in the presence of histamine but absence of extracellular calcium, you'll note that the store-release calcium is similar under both oxygen concentrations. And when we add back calcium, 2 millimolar calcium, you'll see again that the traces are the same. But when we look at influx, you will note that the concentration or the plateau is lower in cells at 5%, mimicking what we showed you in panel A. So clearly, calcium signaling is markedly affected by cells adapted to 5% oxygen. I'd like to focus you now, please, a lot of writing on the slide. Just look at the left-hand slide, please. And what I'm plotting here is the viability of umbilical vein endothelial cells versus increasing treatment of ionomycin, a calcium ionophore, on the x-axis. And what you'll note in the open symbols, the open white circles are cells pre-adapted for 5 days to 18% oxygen versus cells in the black squares in 5% oxygen. The cells adapted with a different phenotype at 5% oxygen show greater protection against ionomycin-induced cell death. I'd like to skip what's on the right-hand slide for brevity. You can pick this up in the article of these papers. I move on now. Complicated slide, but it's going to be very easy. We're very interested in antioxidant defenses regulated by this ubiquitous redox-sensitive transcriptional pathway KEAP1 NRF2. If you look in the panel at the top part, normally under healthy conditions, NRF2 is bound to a partner called KEAP1 and targeted for ubiquitination by the 26S proteasome. That's the left-hand part of the slide. If you modify cysteine residues on KEAP1, you will lead to a conformational change in this complex, which effectively, for simplicity of this talk, allows NRF2 to accumulate in the nucleus where it binds to an antioxidant response element, abbreviated ARE, with a small MAF protein, to drive the upregulation of stress protein, hemoxygenase, NQO1, and glutathione biosynthesis enzymes, GCLC and GCLM. I can't go through the rest of the slides since I don't have a pointer. If you want to read about it, pick up FRBM 2015, volume 88. There are 480 pages of state-of-the-art articles on redox signaling regulated by this transcription factor. But what happens in our cells when we adapt them to 5% oxygen? Here we have data where we're looking at a target of NRF2, hemoxygenase 1, and you will see that in 4 passages or 3 passages along the x-axis, the first 2 columns, the open one is control. The black column is a stress stimulus, in this case an electrophile, diethyl maleate. You'll see the increase in HO1. If you have cells in the second row looking across air, 5%, you lose the response. And if you have cells in the 4th column or look across, whenever the cells see 5% oxygen, this particular NRF2-driven protein or enzyme is not upregulated. There's plasticity in this action. So cells need long-term adaptation to physiological normoxia to highlight this response. We know why this happens, and we can see this here in human coronary artery endothelial cells for HO1 and NQO1. Please look under air. We get a robust increase in the induction of these very important antioxidant enzymes, and we lose that response when cells have been adapted for 5 days to 5% oxygen. So clearly, these 2 NRF2-driven genes are downregulated under conditions that recapitulate physiological normoxia in vivo. Now, we know why this is happening because if I look in this paper, if you look at the ordinate, the y-axis, BAH1 is protein expression, white columns. Please focus just on the white columns in air. You see a significant increase after 5 days and 5% oxygen, where BAH1 is upregulated. Forget the black columns for this moment. Upregulation of BAH, it's a negative regulator of NRF2-driven HO1 expression. So we were able to demonstrate that under physiological normoxia, BAH1 levels are up, and that this particular response is markedly attenuated. In my next slide, I'm showing you some siRNA experiments in the immunoblot at the top. Cells have been adapted, as the bar at the top shows, 5% oxygen for 5 days to physiological oxygen. Note that when we silence BAH1 with siRNA, that's the right-hand side of that immunoblot, that an important glutathione enzyme, also regulated by NRF2, but not downregulated by BAH1, is unaffected. So what we argued in this paper is that in vivo, these two glutathione synthesizing enzymes are what the cell is using to defend itself against an electrophilic agent, in this case, diethyl maleate. And this is the schematic. It's a little bit busy. Under hyperoxia, I'm bringing home now, if you stimulate a cell that has been maintained under 18% oxygen or hyperoxic conditions, you will get NRF2 going to the nucleus, driving these antioxidant genes. But BAH1 levels, shown in the nucleus there in the lower part of the left-hand side, is not inhibiting that pathway. When we adapt cells on the right-hand part of this slide to 5% oxygen, you'll immediately note that although NRF2 goes to the nucleus or translocates, it cannot drive HO1 and NQO1 because BAH1 protein has actually blocked that signaling pathway, whereas the glutathione genes are sustained. If I've gone too fast to you, forgive me for time, but look up the paper Chappell et al. In Free Radical Biology and Medicine 2016. It's highlighted in the slide. And what I'd like to end my talk with is perhaps what I consider the most practical demonstration of the importance of measuring physiological responses, cell signaling, in cells, in culture, at the right oxygen level that they see in vivo. Here we're looking at a graph from a paper of ours and colleagues at King's, where we're asking the question, are pulmonary epithelial cells sensitive to copper nanoparticle toxicity? In other words, if we use the nanoparticles as vehicles to deliver drugs, should we not first establish the sensitivity of cells to their damaging actions? And if you look on the right-hand side of this figure in the middle, you'll see glutathione levels on the ordinate, nanomoles per milligram protein, and the columns are denoting cells that have been cultured in hyperoxia, standard culture incubators, 21% oxygen. You'll note that increasing concentrations, that's moving from left to right in that graph, have no effect on cell viability or glutathione levels, but very high levels, 2 centimeters squared per centimeter, I can't read it, it's too small, actually cause damage. If, however, we take these same cells and adapt them to 13% oxygen, a level they would see in vivo, you will note immediately that glutathione levels are virtually half, and now these nanoparticles are causing toxicity and lowering glutathione levels. Facetiously, the right-hand part of this figure could have gone to Nature Medicine, go to clinic, use the nanoparticles to deliver drugs. But the left-hand side of this figure, done under physiological normoxia for these cells, would caution and say, be careful, this is not viable, the cells are sensitive. So here's a classical example where we can see sensitivity to damaging nanoparticles clearly differentiated if cells see physiological normoxia. Okay, that's not coming in, that's blank. Okay, here it comes. These are publications that you'll be able to download from the papers we've written. I've not looked at many others in the literature. These are the ones that we've validated. They work from the bottom one in stem cell reports in dental pulse cells. The next one up from the bottom in Febb's letters is in epithelial airway cells. The next one is our work in umbilical and coronary artery endothelial cells. The next one is in endothelial cells related to NO. The other one is CIRCA. And finally, I'm illustrating the top. This is an invited physiological review article currently under review in the last 2 weeks. It's 130 pages long, and it'll give you the oxygen concentration in virtually every tissue, every organ in vivo. And that's an article by Tom Keeley and myself my postdoc. Like my colleagues before me said, none of this would be possible if I didn't show you the people. These are the happy people that have partly contributed to this work. British Heart Foundation have supported it. A net grant from heart research has been absolutely critical. And without the interaction and the support in kind through R&D from Baker Ruskin, BMG LABTECH, and Luxel, none of what I just shared with you would be possible. At the top, you'll see myself, not because I'm old and wise, but Richard Seow has played a critical role in this, as has Ron Jacob, Sarah Chappell, and Thomas Keeley. The others are colleagues in our group, and there's a lady down below highlighted called Gabriela Warpsinski. She's looking at brain endothelial cells, ischemia reperfusion injury, again, applying some of these techniques. And because I always like to end on a good note, for those of you interested in redox signaling, I strongly encourage you to enroll in this international conference on free radical research, which will be held in Lisbon, 4th to 7th of June 2018. The URL is there, and we will be running a oxygen physiological normoxia, hopefully symposium, and hopefully having a plenary lecture which will underpin what we've just shared with you. Thank you very much.
Thank you for your presentations, Giovanni, James, and Catherine. It is now time for the Q&A. This is your opportunity to ask all the speakers your questions. To ask a question, all you have to do is type it in where it says ask a question and then press submit. So our first question asks, how long can a kinetic cell-based assay last using the CLARIOstar? Catherine.
Okay, thanks, Joshan. So you may have seen from the data I presented today, we had 72 hours of data collected in the Promega assay. I personally have seen assays go out to 4 days, but that would be very much in the hands of the cell type and what the user was looking to do. Of course, you know, in terms of culture conditions, normal culture conditions as well, in terms of when you might wish to change medias, etc., etc. So, but we certainly can do proliferation assays lasting at least 72 to 4-day assays.
This is James here. I can come in with a little bit of additional information there. We would have done long-term oxygenation measurements in that regard, and the fact that CO2 is maintained allows this to be done, and it's a pretty simple thing to add a breathable seal to the top of the plate to minimize evaporation, and that really does allow you to extend measurements for a number of days.
Excellent. Thank you both. And our next question is actually for you, James. It asks, what fluorescent wavelengths are the MitoXpress compounds? Do you have to make different options for different fluorescent needs?
We do have a range of probes, but the ones that we find work best on plate reader platforms are all from a very similar family. So they would typically excite around 380 nanometers, and we would be measuring emission around 650 nanometers. And what that does is it allows us to overlay these probes with other readouts, which I tried to address during the talk. So we've successfully multiplexed with things like JC1 or our own MitoXpress-Intra, or other probes that inform on biology but can be multiplexed in the same well at the same time, so that you're recording all parameters from the same cell population.
Excellent. And our next question, I think, goes to Giovanni. Is physioxia, so 5% of PO2, suitable for all types of normal cell lines?
Very good question. The answer is no. What is very important is that by understanding the in vivo oxygen level in the given tissue, the heart, the kidney, the lung, wherever you are in the human body, that will dictate the level of oxygen that you may begin exposing your workstation to and then your cells. The second thing that's critically important is if you're doing a static monolayer of cells or a cell lining culture in this workstation, is the diffusion of oxygen, as I tried to highlight in one of my slides, that you might actually have a gradient. And if, as James showed in his very elegant thing, though I don't know if the movie was running, if you had a cardiomyocyte that was contracting, and stimulate it, for example, with isoprenolone or isoproterenol, as he showed in his slide, that would use oxygen. And without a blood flow, as in vivo, you could be using up the oxygen through this unstirred layer and maybe actually have the intracellular oxygen level too low for what we would call physiological normoxia. And in the review article, hopefully when it's published in Physiological Reviews, you will be able to follow from lung through different organs in the human body, including chondrocytes, kidney, what have you, what oxygen levels have been measured using microelectrodes in vivo. And we use that as a basis to inform what we do in vitro. It's a long-winded answer, but the answer to the question was no. You have to be careful and refer it back to the level measured in vivo.
Excellent. Thank you, Giovanni. And our next question asks, is top or bottom reading better for cell-based assays? Catherine, is that something you can answer?
I guess that's one for me, quite simple. Whenever reading cell-based assays, particularly in monolayer, we would typically recommend reading from the bottom. The air optics in the CLARIOstar ensure the best performance in cell-based assays, and we can actually use our Z height adjustment to actually focus in on the layer of the cells. So bottom reading for cell-based assays.
Excellent. Thanks, Catherine. And our next question is for Giovanni, and this one asks, in the 4-2-am calcium uptake assay, to what was the calcium uptake normalized to? Was it normalized to cell number?
This is an important question. I didn't have time to describe it in sufficient detail. These are endothelial cells plated into 96-well plates. So these are population of cells loaded with FURA2, and what we're looking at is the population fluorescence signal in one of the slides I showed you. So we're not looking at single-cell calcium, but we're looking at a population, and we're referencing that to the baseline level. Yeah? You could ask questions, do you have the same amount of loading, etc. But in this case, because we're looking at a large population of cells, we're getting the average change in stored calcium release and influx. I'd like to correct something. I made a minor mistake in one of the slides I showed you in the calcium add-back experiment, where basically what we demonstrated is not that when we added back calcium in the presence of histamine, we saw no difference in influx of calcium between HUVEC, these are endothelial cells, cultured in air, or 5%. So if you go back to the slides and download, that was an error on my behalf, and I apologize for that.
Excellent. Thanks, Giovanni. And our next question asks, is it possible to measure PO2, PCO2 or pH in 3D micro-engineered organ on a chip, such as lung on a chip? Who would like to answer that question?
I can take that. So this is James from Luxel. The measurements are feasible, particularly if the chip can be mounted on the plate reader. So lots of these can be measured in stop flow on holders on plate readers, and if that's possible, then essentially the same protocol that we described during the talk is possible, either measuring pH or oxygen concentration. CO2 is a more complicated measurement, and that would not be as straightforward, just purely because how those optical measurements are typically performed. But we have done measurements on various 3D structures, and we can assess oxygen concentration on the surface of those structures and also within those structures, and that can be quite illuminating. So from the plate reader's perspective and the probe perspective, it's simply a matter of being able to mount it on the reader for interrogation.
Excellent. Thanks, James. And our next question asks, for long-term assays in the microplate reader, does evaporation play a major sort of issue since it's not humidified? Catherine, is that something you can answer?
Yeah, so I think James already mentioned the availability now of breathable seals. There are also microplates available that offer channels to fill an area surrounding the wells to kind of give kind of an added humidity, if you like, feel, provide kind of a humidification to the process. So certainly in those longer term trials that we talked about, we haven't seen a major impact of evaporation. It performs very similarly to having your plate in the incubator from what we've seen by sort of weighing plates in and out of the systems at different times. We also, the way the CLARIOstar heats the microplate with a heat plate above and below the microplate creates like a sandwich of heat around the plate. And that top plate is slightly warmer by about 0.5 of a degree to kind of minimize those evaporation type effects. So for what we've seen, certainly in the trials we've done, users are typically able to use the full plate without seeing evaporation as an issue over time.
Excellent. Thanks, Catherine. I think we've got time for just one last question. And this one asks, it was shown that intracellular oxygen was highly dynamic depending on many parameters. And the question is asking, how can we control physiological oxygen if it's constantly changing? Who would like to answer that?
I can take that, and then maybe some people want to add to it. I think there are two aspects to this. Just looking at control, yes, it is dynamic, which is an important thing to realize, and it's more that metabolism can be dynamic, and then by extension, oxygenation can be impacted. But I think you can at least be working within a physiologically relevant range, for want of a better term. But I think what's also important is that when you're studying the relationship between oxygen and other parameters, that you know what the cells actually experienced, because that could be an important input. And Giovanni shows some nice data looking at the impact of oxygen concentration on HIF stabilization. And if you were to assume that the oxygen concentration applied was what those cells experienced, your interpretation of those data would have been significantly different as compared to knowing what the cells actually experienced, so that you were establishing the correct, accurate relationship between oxygen concentration and, in that instance, HIF stabilization. And again, that can have pretty profound implications on how those data are interpreted.
I'd like to concur with James. I think it was a very good answer there. Clearly, I would like to come back to the concept that if you're working with static monolayers of cells, wherever they come from in the human body, even if you set the right physiological level of oxygen in the workstation, the Baker-Ruskin workstation, if the cell is metabolically very active, I didn't have time to show you slides, it will clearly depend on whether it's a contracting cardiomyocyte or a HeLa cell or an endothelial cell. The HIF story is important, and I'd like to bear out one final point. You cannot just simply take cells from air into a lower oxygen level for 4 hours, 6 hours, 10 hours, 12 hours, 24 hours, and assume that you've reset that oxygen phenotype. You haven't. We have clearly shown you need at least 4, 5, 6 days of continuous culture. Now, yesterday when I gave a seminar at St. George's, or the day before, a question was immediately asked: what do we do when we have contracting human cardiomyocytes that we cannot maintain for 5, 6 days in culture? There are different problems encountered in looking at that cell type. But please bear in mind that it needs long-term adaptation and that the process shows plasticity.
Excellent. Thank you both. Well, I'm afraid that is all we have time for today. I would now like to thank Giovanni Mann, Catherine Walk and James Hines for their presentations and for answering your questions. I would also like to thank the webcast sponsors, BMG LABTECH, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you will join us again soon.